Good evening, namaskar to all. And today I'll be teaching you Krebs cycle. This is the second step of respiration, and that's a part of 11 NCRT book. And here you see in the Krebs cycle, this was given by Hans Krebs. That's why the name is Krebs cycle. And here this pyruvate or pyruvic acid, which is the end product during glycolysis, that will be taken. And here you see this pyruvate is a 3-carbon compound, and in this pyruvate a process will be occurring called as oxidative decarboxylation, and one coenzyme A will be added. You see here this decarboxylation, D means removal. Here one carbon dioxide is removed, you can see here. And whenever one carbon dioxide is removed, one carbon gets removed, and this compound, by the addition of coenzyme A, here a 2-carbon compound called as acetyl coenzyme A is formed. That's a 2-carbon compound. And in this step, NAD plus will be converted into NADH and H plus. Now this acetyl coenzyme A, which is a 2-carbon compound, that will be added to oxaloacetic acid. That's a 4-carbon compound. And this 4-carbon compound oxaloacetic acid will be added to acetyl coenzyme A in the presence of citrate synthase. That enzyme will regulate the production of citric acid. This citric acid is a 6-carbon compound. Here oxaloacetic acid is 4-carbon, acetyl coenzyme is 2-carbon compound, and together that will be 6-carbon compound. As the first stable product here, citric acid, so this cycle is also called as citric acid cycle. And here in the citric acid, 3 carboxylic groups are there. So it is also called as tricarboxylic acid cycle. Now this citric acid, which is a 6-carbon compound, it will undergo decarboxylation, and one CO2 will be released, and that will be forming alpha ketoglutaric acid. Now here this is a 5-carbon compound now, because one carbon is released, and here in this step NAD plus will be converted into NADH and H plus. Now this alpha ketoglutaric acid, again it will be undergoing decarboxylation, it will be forming succinic acid, which is a 4-carbon compound, and in this step NAD will be converted into NADH plus H plus, and one GDP will be converted into GTP, and this GTP energy is equivalent to ATP. Now this succinic acid will be converted into malic acid, and in this step FAD plus will be converted into FADH2. Flavin adenine dinucleotide will be converted into FADH2. Now this malic acid will be forming further oxaloacetic acid to continue the cycle, and in this step NAD plus will be converted into NADH and H plus. Now the cycle is completed here. Let us see how many NADH, how many FADH2, and how many ATPs are produced when one pyruvic acid undergoes Krebs cycle. Now here pyruvic acid plus, in 4 places you see, here 1, and here 2nd, this one 3rd, and here 4th. And 4 NAD plus are taken, plus 1 FAD plus, then 2 H2 are taken, plus 1 ATP plus inorganic phosphate. So that will be converted into four NADH plus FADH2 plus ATP. Now you see here, total, let us calculate how many ATPs are used. 1 NADH is equivalent to 3 ATPs. So here it will be 3 into 4 ATPs. Now 1 FADH2 is equivalent to 2 ATPs. Now here plus 2 into ATPs plus another ATP, that is ZTP equivalent. So here like this, 15 ATPs in total produced by one pyruvic acid when it is undergoing Krebs cycle. Actually 2 pyruvic acids are produced. So it will be, whenever we will be taking 2 pyruvic acids, so it will be 30 ATPs. Now here you see, this is for 2 pyruvic acids or 2 pyruvate. Now during this glycolysis, gross ATP production is 8, and now during Krebs cycle, gross ATP production for 2 pyruvate it is 30 ATPs. Now 30 ATPs plus 8 ATPs, it becomes 38 ATPs. So whenever one glucose molecule undergoes oxidation, so it produces 38 ATPs in aerobic respiration. Thank you very much.